Okay, so let's talk about T cells. And kind of like this cartoon is showing you, so here's we have a dendritic cell and it's trying to present this MHC complex to this CD8 T cell, or as we're going to see, this is going to be a cytotoxic T cell. But as I was mentioning, not all of them recognize each one. So you're going to have to find the specific one that recognizes it. So it's like, oh, it's making me so sad. But I'll move on to find one that does recognize it. Okay. So at any given time, most of your T cells are inactive. The way that we activate, first we'll start with the activation of helper T cells. It'll be a little bit easier to think of it in that order. The antigen receptors on the surface of T cells are called T cell receptors. And these are what will bind to the MHC2. Okay, again, we're talking helper T cells. So remember, MHC2 are on the surface of antigen-presenting cells. And then, again, millions of these different T cells, each will have a unique T cell receptor, which will respond to a unique epitope. There's also another protein on the surface of these cells. It's called a CD protein. And then whether it's a CD4 protein, we'll mark it as a helper T cell, or a CD8 is a cytotoxic T cell. And that's basically to help maintain that receptor MHC coupling. But again, this helper T cells will have a specific CD4, and cytotoxic T cells will have a specific CD8. You do need to know that the CD4 goes with helper T, and the CD8 goes with cytotoxic T. They are often referred to by these numbers and not their full names. Okay. A T cell will become activated if it binds to that antigen MHC complex and receives co-stimulation, right? Then that co-stimulation is important so that it doesn't accidentally get triggered, because if T cells are triggered, that can cause death to other cells in the body or autoimmune diseases. So you try to prevent that from happening by having this sort of dual authentication, if you will, in the body. And then different co-stimulators will affect T cells in slightly different ways. But that's what you're seeing here. You have an antigen-presenting cell. You have the recognition as well as co-stimulation. It activates. If it recognizes but there's no co-stimulation for whatever reason, it will not recognize it, and that T cell actually will usually die in that case. Because something's wrong. If it's not activating and it recognizes it, then something's wrong with that T cell. Okay, but let's say that helper T cell bound okay to that MHC2 on the antigen-presenting cell, and it activates, right? It goes into the active form. So again, it will do the same thing that the B cells did. It will start dividing and differentiating into an active form and a memory form. The memory form is the inactive, there for another day, right? And that co-stimulation that it's going to deliver is so important. That's going to be via something called interleukin-2. And you can see here, caps, very important. Pretty much that interleukin-2, again, even though B cells are going to be able to do things by themselves, this acts sort of as a major stimulant, allowing them to work even faster and better. And the cytotoxic T cells absolutely have to have this interleukin-2, otherwise they will not activate. Again, helper T cells display CD4. As I mentioned, they become the active and memory form. The memory is inactive. They're there for the next time you might face the same infection. Now, on the other hand, cytotoxic T cells, remember, these are going to kill off infected body cells. So these have that CD8 protein on their surface, and they are now going to recognize MHC1. Remember, all cells of the body, except for red blood cells, will have an MHC1 complex. So these are things like body cells that are infected, tumor cells, transplanted cells. So like this image indicates, here's an infected body cell. It underwent the endogenous presentation of the antigen, right? It's infected, so it took at least one of those infected microbes, viruses, whatever, put it onto that MHC1 and said, hey, I'm infected, come and kill me. this cytotoxic T cell has bound to it and says, I am absolutely ready to kill you. That helper T cell says, yes, you know what, I've been shown this by an antigen presenting cell. That is the thing that we're here to kill. I'm going to give you that co-stimulation. That cytotoxic T cell says, great, I will now kill you. And then once it receives that co-stimulation, not only will it kill that infected body cell, but it will also activate, divide, and again form the active and memory version of it. Okay, so this is just what I mentioned. So it needs the co-stimulation to become active, as well as to kill off the infected cells, as well as form the active versions, as well as memory versions. And each one of these cytotoxic T cells can go on and kill multiple infected cells. In the way they do this, it's one of two different ways. They either use granzymes, and the granzymes will enter the infected body cell, cause the cell to burst, releasing the microbes so they can no longer hide, in which case macrophages can come along and gobble up the microbes. Or they use the perforin granulysin combo, where they poke holes, send in the granulysin, which kills both the cell as well as the microbe. So in this version, it does both. In this, it simply releases the microbe. So the summary of our T cells: helper T cells will secrete many of these cytokines. The most important one that we're going to care about is interleukin-2. That acts as a co-stimulator for other helper Ts, cytotoxic Ts, as well as B cells. These cells display CD4 protein on them. Cytotoxic T cells have CD8 protein and will attack either pathogens and infected cells. And then the memory version of either one of these two is the inactive form, but it is there for the next time you get infected. And then as far as the cytokines go, there's many different versions of these, but basically they are going to help cells, stimulate cells to divide or to grow. We've seen perforin as an example, granzyme as an example, interleukin-1 is an example, as well as interleukin-2. So this is just a summary slide. And these, depending on what thing it is, it can be secreted by a variety of different cells. Okay. I'm going to go on to, hopefully that made sense. I'm going to go on to one last slide because it kind of goes with this section. The ways we can acquire immunity. We can either acquire it naturally, meaning you get sick and you respond to the antigen. It can be artificially acquired active. This is through the use of vaccines, so that's not natural. The vaccine itself is artificial. However, you are still developing your antibodies and naturally responding to it. There's artificially acquired passive, means so it's artificial, so it's not happening naturally, and it's passive, so you're not producing any antibodies. So this means you're going to simply take antibodies from one person and inject them into another. Those antibodies don't live very long, so this isn't a long-term solution. And then there's naturally acquired passive, so it's natural, so you're not artificially inserting anything. So this occurs when antibodies are passed from mother to the fetus. However, it is passive because, again, the fetus itself is not producing the antibodies, it's just getting it from the mom. So again, if you're not producing antibodies yourself, you're receiving it from somewhere else, that's passive. If it's via an injection, it's artificial. If it's natural, again, that's just mom to fetus. Artificially acquired active is the vaccine, but you're still actively producing antibodies. And then naturally acquired active is when you simply get sick with something. Okay, I will end that there, and hopefully this all made sense to you, but if not, please feel free to email me with any questions.